the flower probably would sorry would probably at least put pollen on the stigma, if not affect pollination. I guess I have to. Okay, so, and it does have beans, as coral bean implies, but they aren't always coral. They typically are red, but there is actually quite a lot of color polymorphism in the beans, as well as polymorphism in size. And just like you can write on the calyxes to keep your flower straight, you can also write on the seeds to keep your seeds straight. So when I tally the seed set, you can see in this opened pod, as in most legumes, the ovules are held on alternate sides of the pod once it splits open. And so you can actually, when the fruit is dry, pick open the pod and you can score. There's an ovule at the very top right that didn't get pollinated. So the 2 is the second seed in the bench. Then on the right side again, you can see there's another non-pollinated ovule. And then, so I can basically score the seed set and the fate of all the ovules that weren't actually fertilized by opening these pods. And actually, I brought those seeds. If you want to see them in person, there's a box of seeds. See what you're missing if you didn't come to this talk in person and are watching on Zoom, you don't get to see the seeds. So, okay, so I was very lucky to be able to study, I am very lucky to be able to study coral bean in 2 amazingly beautiful environments. So in addition to it being a lovely plant and having wonderful pollinators, you get to hang out in really wonderful places while you do your work. This is my initial study site in the Horseshoe Canyon in the Chiricahua Mountains. And as I told you earlier, there's this channel of giant rocks that is inhabited by a population of Erythrina, the big one in the left of the shot, but you can also see several additional plants all the way up this river of rocks. And this represents a population that I've had under study now for almost 15 years. The other site where I've been working is at Texas Canyon on the grounds of the Amarin Foundation, which is an archaeological foundation that's been very generous in helping me do my research. And this plant you'll see in other slides later on. This is plant 1342, and it looks like a whole bunch of plants along this crevice, but it's actually a single plant with a single caudex traversing this crack. If you go a little bit, a foot further, then you would fall off a short cliff, and that would probably be painful. But the point here is that this plant is very high, it's highly elevated from the rest of the population, and it's a real magnet for hummingbirds. The other point I want to make with this slide is, again, as in horseshoe, these plants are not on their own in the world. They're parts of populations. And so most of the rest of the stuff I'm going to talk about today is driven not so much by just saying, oh, here's a plant and here's how big it is and here's how many flowers it has, but trying to understand the population biology, the kinds of phenomena, patterns, and processes that go on in a population setting. And evolution occurs in populations. Jack, is there any way we can get rid of the stuff at the top of the screen so people can see what is at the top? Can we? Hide video panel. Hide floating meeting controls. There we go. Woo! This is actually surprisingly difficult to hold this microphone and talk to Zoom and you guys as well. So forgive me, but okay, thank you. Okay, so the point is that evolutionary processes take place in populations. They don't take place in individuals. And so my interest is really in the kinds of phenomena that go on in a population over time that make that population persist into the future. So I'm very interested, especially in processes of gene movement, of pollen flow between individuals, and of the genetic consequences of that. But I'm also interested in the floral biology and the kinds of mechanisms that this species has evolved that moderate how pollinators move between plants, that moderate how herbivores might spread between plants. And so the point here is simply that we're going to be talking about population processes, things that go on in, hello, populations. Oh, there. Okay. So this is just a sort of a didactic slide about the kinds of things that population ecologists address. Maybe I have to do this for every panel. Okay. Population ecology, population ecologists look at a whole bunch of different features of things that are really present only in populations. One of them is demography. You can't have a population size without having obviously more than one plant. So recruitment, growth, death, longevity, age are all components of that. But reproductive biology is one of the factors that I'm most interested in. That includes the biology of the flowers, how long they stay open, how many open at a time, the timing of flowering in general, gender expression and nectar. You can see the list of various characteristics that go along with trying to understand the reproductive biology of a population. In addition, I'm going to talk a little bit about ecological factors, interactions with other species, and in particular, in this case, with interactions with herbivores and florovores, which are a component of the population biology of Erythrina. And I'm extremely interested in the genetic components as well. I'm not going to talk as much about that because I'm still in the process of doing genetic analyses and I don't have a lot of my data yet to talk about. But the long-term goal would be to try and understand which plants are reproducing, what genes they're putting into the next population, and what that might say about the future of the population as a whole. So here's another view, maybe not a very good one, but another view of a population. And this is an aerial photo of my Amaranth site. And so right here in the middle, right here is plant 1342, the picture I just showed you at the ledge. And as you look out from plant 1342 in this direction, you see what we saw in that photo. You can see the road in the distance. You can see this big bare spot here, which is where a giant oak tree got incinerated by the fire last summer. And so now it's a pile of ash. And within the view of this plant from this perspective, there are probably about 60 Erythrina plants, most of which are part of my study population. So I've put in little markers to show you what a population looks like. And part of this is because I like to think of population biology as being in some ways a choreography of the population. This is what the population looked like on the day I mapped them. But if I go back a year later, some of those plants will have died. Some new plants may have recruited, and not only will the physical distribution of plants be different, but the population that a pollinator will encounter is going to be different. There's going to be a sort of a long-term choreography, a time-lapse photograph that population biology can help you understand. And it's that dynamic that I find the most fascinating about plant populations. They're not like animals. They don't move around every day. They move much more slowly, but we can interpret their movements with more grace, I think. And here's that same population. You can see here's this curved section here, and then a strong central port, and then this clot down here. This is the same population where I've scored each plant for its genotype at a microsatellite locus. This is a single microsatellite locus. And so plants that are marked with two different colors are heterozygotes for two different genetic loci. And this just sort of gives you now a genetic picture of what the physical structure of the population looks like. And it's this then that will let us do paternity analysis and track genetic movement in the population. So this is kind of a messy just diagram of the kinds of things that play into the questions I ask in a population. Breeding system, it's reproductive biology. Demography, genetics, and environment are sort of functions of the population. And then each individual in the population is interacting with pollinators, herbivores, competitors, and you really have an ecological and evolutionary web that works itself out through time. And those are the kinds of questions that I think are most interesting. So to start talking about reproductive biology, I want to just use this rather verbose slide. David Neal in 1987 published a treatise on Erythrina breeding systems. And this particular list is focused on species in the section Erythrina, which includes Erythrina flabelliformis. And this was his assessment of the breeding and floral qualities that he thought were characteristic of the Erythrinas in his study group. And so some of these are not under discussion. Red or orange flowers, that's certainly the case for Erythrina flabelliformis. But I wanted to test Erythrina flabelliformis against all these other traits. And so tubular flower shape, check. Flowers oriented outward, check. But some of these other qualities are things that I tried to address experimentally, and I'm going to talk to you about in a little more detail. The ones that are highlighted in purple are the things that I'm not sure were true of Erythrina flabelliformis. And these are factors that are going to influence the breeding system. Flowers per tandris means that a flower produces pollen before it's receptive to receiving pollen. So it's functioning as a male before it becomes functional as a female. And David suggested that these flowers were male first on the first day, and that only on the second day was the stigma receptive. That kind of separation between male and female phase is important because it minimizes the chance of self-pollination. So it means that birds that visit the plant on the first day may remove pollen and take it to other plants, other flowers, but they have to visit again to actually fertilize the plant itself. He also suggested that in his species, the style actually grew on the second day, elongated so that it was no longer embedded in the anthers, but was actually extended beyond the anthers, again, to facilitate pollination. Copious nectar, he said, was produced over 2 days, which would facilitate birds coming to the same flower more than once. And he argued that the plants were self-compatible, but mostly outcrossed because of these mechanisms that would minimize selfing. And he also suggested that in this section of Erythrina, the only legitimate hummingbirds that would set fruit would have bill lengths greater than 28 centimeters. Now that's actually a challenge because there aren't very many hummingbirds in Arizona who have bill lengths over 28 centimeters. So the question is that actually the case for my plant species or not? So we did a bunch of experiments to try and understand this. We looked really closely at the plant flowers, and we asked questions about when flowering started, how fast flowers opened, how the structures inside the flowers moved, and so forth. And so I'm mostly just going to give you the take home for what we found. One point I want to make is that these flowers of Erythrina are not out there on their own. Just like a population is made up of a lot of individuals, and inflorescence is made up of a whole bunch of flowers. And so although flowers will have individual traits that will make them more likely to be pollinated in a certain way, the fact that these are aggregated in an inflorescence is an important aspect of its pollination. These are flags. These are giant flame colored signals to hummingbirds advertising the location of a plant. And so the aggregation of flowers into an inflorescence is one of the factors that's a part of the breeding system of this plant. It means that it attracts birds from further away. A group of inflorescences such as these creates a challenge for the plant because if that hummingbird moves between flowers in this photograph, it will actually be moving between flowers that are the same genotype. These are all the same plant. And so that would affect selfing rather than outcrossing. So there's this contrast between whether or not you're drawing in lots of birds to visit lots of flowers on a single plant, or whether by having only a single flower inflorescence available, you're basically forcing the bird to fly away and take your pollen to another location. Now, obviously, that's not something the plant is thinking about, and it's something that happens as a plant grows, as it gets bigger, as it becomes smaller. So those are changes that happen in the life of the plant, but they affect the way that pollen is going to move around in the population. If you look closely at an inflorescence, what you can see is that the flowers are produced in fascicles of 3 stems. So 3 flowers come out at one point on the stem. And those flowers usually all bloom the same day. So you have a whorl of flowers usually that opens every day as new flowers. Flowers last 2 days. And so the next day, those flowers are now second day flowers and a new whorl opens above it. So this means that there's a constant progression of new flowers being produced as you move up the stem, and there may be up to 20 flowers open on an inflorescence in any one day. This photograph is my colleague Alberto Burquez, and he's helping me with some nectar work in this slide, but he is also a colleague who's been a big part of this research, and so I just wanted to mention him as we go past. So a plant can have, as you can see, up to 80 inflorescence, up to 80 flowers per inflorescence, which means if it puts out 10 to 15 flowers a day, it can bloom for at least a week and probably several weeks. And the flowering rates differ in different plants, but individual inflorescences can flower for up to 3 or 4 weeks. And the plant itself may flower for 1 to 5 weeks. Or if it's a small plant, it may have only 2 inflorescences and be done in a week. So every inflorescence contains up to 80 flowers, and each of those flowers has multiple seeds. And so every inflorescence is actually an investment, and loss of inflorescences to herbivores is potentially costly to the fitness of the plant. I'm going to make this go forward. Okay, thank you. Each individual flower has 10 to 14 ovules, but it's also the case, at least in my populations, that many of the flowers do not set fruit. And most of the fruits that are produced are not fully pollinated. And so there's a question that could be asked about this, and it's not always the case, but is this because there aren't very many pollinators? Is it because the pollinators that are visiting, the smaller birds that we have here, are ineffective? Or is it because of the fruits, which are actually choosing, in a physiological sense, to keep flowers that have been outcrossed and to selectively abort flowers that may have been selfed? And for that, I don't have any evidence for this plant, but it has been demonstrated in other plants. And so that's not outside the realm of possibility. So the numbers mean that plants are capable of producing a huge number of seeds, but in fact, in my populations at least, they don't necessarily do so. Now, in the observations we've made in Mexico, the flowering is roughly of the same sort of patterning, but you do see a lot more seed set on some of the plants in Mexico, and they have a lot more visitation. So there may actually be a dearth of hummingbird pollinators at this point, at this place in the distribution of this plant. Nectar is the reward that the plant offers for pollination. And so in this photo, I've torn away the floral tube, and I've got a calibrated micropipette inserted into the flower under those little wings to gather the nectar. And you can measure the length of the nectar. You can see the meniscus. You can measure the length of the nectar in that tube and quantify the nectar production by these flowers. And nectar production is a crucial element of attracting the hummingbirds. Obviously, that's one of the reasons they're there. They're not there just because they think it's a pretty plant. So the questions that Alberto and I particularly discussed or investigated were patterns of nectar production in open flowers. The first question, how much nectar does a plant produce? In a single flower, in a first day flower, the plant can produce between 10 and 30 microliters of nectar, which is not a vast amount, but it's a reasonable drink, especially if the bird is visiting several flowers in sequence. How rich is the nectar? The concentrations are about 20 to 30 Brix, which is gram sugar per gram solute, so basically percentage. And that's roughly what you would expect for plants that are hummingbird pollinated. And how long? Remember that Neil suggested that there was copious nectar being produced over 2 days. In our studies, we found active secretion mostly only in the morning of the first day of flowering. And so in this case, our observations are actually different from what Neil suggested. And in fact, there were several points of his hypothesized breeding traits that we found not to be the case. We did crossing experiments on first day flowers and second day flowers using both the pollen and the ovules on flowers of different ages. And what we found was that flowers are not necessarily protandrous. The pollen is present on the first day clearly, but it's also still effective in producing fruit set on the second day. So there is no temporal separation between male and female functions. The stigma is not only receptive the second day. I could pollinate first day flowers and I would get fruit set from them. So the stigma was actually receptive at the beginning of the flowering period. The style does not show any evidence of elongating. And although the style position is variable between plants, I measured various changes in style length between day 1 and day 2 flowers. And in some cases, the anthers actually extended beyond the style. So the style does not uniformly extend. And although there's nectar produced, it's not produced over the whole 2 days. The nectar is mostly concentrated on the first day. The plants do seem to be self-compatible. I was able to get self-compatible crosses. But in fact, when we've done the genetic work on a few of our plants in the studies we've done subsequently, it is true that there's mostly outcrossing. In 44 seeds that we sampled from a couple of plants a couple of years ago, there was only 1 seed out of the 44 that was potentially self-pollinated based on the number of gene loads that we had. So somehow the plant is avoiding selfing, even though it doesn't seem to have the temporal variations that Neil suggested. And then the question of legitimate pollinators and the bill lengths. Well, as I suggested, these are the only 2 birds in our fauna that are here consistently, the blue-throated on the left and the rivolis on the right, that have bills that are anywhere close to 28 millimeters long. The birds that I see at my horseshoe site are broad-tailed, and I've showed you this picture before of a broad-tailed hummingbird visiting the plant. And what you can see is that the hummingbird is pushing its way under the flower. So it's kind of sliding in to get its short bill to where it can get to the nectar. But I think that what it's doing in the process is jarring the stamens in such a way. And because he has to hover to maintain his position, he's doing a fairly efficient job of pollinating this plant, even though he's not traditionally as big as he should be. Another of the species that visits this plant in horseshoe is the black-chinned, again, a small species. And then blue-throated mountain gem and the rivolis, which are the big birds. And I have to tell you, in the time I've spent watching hummingbirds at my study site, I've only seen a rivolis once. I've seen a lot of broadtails, but not very many rivolis. And yet you get really good seed set in some years in these locations. And so it seems like the small plants are able, the small hummingbirds are able to effectively pollinate this plant. And you can see that in some cases, given that there are 10 to 14 ovules, the seed pod on the left is pretty well pollinated. And all of these have, at least half of their fruits fertilized. So the seed set is taking place, although the fruit set is not always very dense. At the same time, it can be very extensive. And this is also in the hummingbird in the horseshoe site. And this is a year where there may have been some hummingbirds out there. I wasn't out there every day and I wasn't watching constantly, but there's a lot of plants that are being pollinated in this setting. And so clearly the birds that are there are able to do the job. On the other hand, there are other situations in which there's very low fruit set. And these are some flowers, some really long flowers at our study site at the Amarand. The plant on the right, the one that still has flowers on it, has actually been flowering for 4 weeks. You can see a little pink mark, a green mark, a blue mark, and a white mark. We marked the flowers every week at the point where the lowest bud was still there. So we know exactly where along the stem a fruit was produced. So we know that the fruit that was there was produced between the day we marked green and the day we marked blue. And so we can track the potential parents in that population as a result. But that flower has been flowering for over 4 weeks, and it has one fruit to show for its efforts. So there's a lot of variation between individual plants and between years. And that's really an interesting observation, because you normally think of fruit set as being one of the central elements of an organism's natural history. Once you set a fruit, however, you're not done. Things will eat it. And ground squirrels and rock squirrels are one of my several nemesis in this situation. They tend to crawl up the plants, and they just willy-nilly clip off inflorescences. They don't eat them, they just kill them. And it's not unlikely, and it happened to me this summer in my censuses over at the Amarant. There wasn't much flowering this summer anyway because of the fire last year, but when I went, I found these evidences of flowers that had been formed but were not going to make it to the end of their lifespan. So there's a lot of herbivory. There's herbivory by mammals, and there's also a lot of herbivory by insects of various sorts. There are seed predators. There's a specific moth I'll show you a picture of that attacks the seeds. There are various insects that consume the flowers and web the inflorescences and essentially kill all the flowers on a developing inflorescence. And then there are also leaf predators. But there's an answer to that, and that is that Erythrina flabelliformis has extrafloral nectaries. And this was something that was really sort of totally unexpected when I started working on this species. But extrafloral nectaries are nectaries that are physiologically on the plant, but they're not inside the flower. We think of nectar as being a floral reward. We think of the nectaries as being inside the flower. Extrafloral nectaries are born on vegetative components of the plant. They secrete nectar, and the traditional explanation and the most general visitor to extrafloral nectaries are ants. These are not uncommon, but they're not something that you may have heard of if you are not sort of a pollination wonk and haven't read some of the literature. But they're very, very interesting, and they represent a potential for recruiting ants to act as anti-herbivore devices for the plant. There are also nectaries on the leaves. That's not uncommon among the legumes. Lots of legumes in different genera have extrafloral nectaries, but they're visited by ants, especially when the leaves are small. And so hypothetically, the ants could be assisting by, attacking caterpillars or leaf bugs or miners or other things to avoid leaf loss. And there's also apparently nectar secretion on the fruits. There's no dedicated nectary. There's no sort of structural nectary on the fruits, but you can see the aggregation of ants along the fruit length. And so apparently there's also some sort of nectar being secreted as well in this situation. So there's apparently an important interaction between ants and extrafloral nectaries that we felt was worth investigating. In particular, we were interested by the presence of nectaries on the flowers, because nectaries on the calyces are not as typical, and they suggest that recruiting ants to the developing inflorescence is an important component for fitness for this plant. And so this is the part of my talk I subtitled Ants in Your Plants. And we investigated a series of questions trying to document this, demonstrate it, and understand what its impacts were for the plant. Now, this wasn't an unknown phenomenon. Actually, Wade Sherbrook and his colleague Joe Sheerans back in the 1980s, I believe, first documented extrafloral nectaries on erythrina and actually did some liquid chromatography and documented the sugar content of the nectar and so forth. And they speculated that there was a fitness component, but they didn't really demonstrate it. We tried to do this. One of my students, Galen Priest, took this on as his senior thesis. And so the year that we were working, we actually had quite a lot of herbivory resulting in this kind of damage. These are what we called webbings of inflorescences. And remember, I told you there could be 80 or 100 flowers on an inflorescence. So if one inflorescence gets taken out of commission by this kind of webbing, there's a significant potential fitness effect for the plant. So webbing of flowers and florivory by potential herbivores is what we assume would be the motivation for the presence of extrafloral nectaries on the sepals, on the calyx. But there are also seed predators that attack the seeds. This is a pyrrolid moth that we raised from the infested seeds, and it's in the literature widely as an erythrina specialist. Well, actually, I think it's kind of general, but it's typically found on erythrina throughout South America and North America on erythrina populations, and it's a seed predator. And the seed predation can be extensive. So even if the fruit looks like it's developing properly, if you wait until the fruit is actually ready to open, it's not uncommon to have this kind of frass and holes. And you can see in this pod, the first 5 or 6 seeds have been consumed by the seed predator. So, simply setting fruit doesn't get you very far in terms of reproduction if you're losing sometimes 40 to 50% of your seeds to a seed predator. There are also foliage caterpillars that attack the leaves. And these are probably not deterred by the nectaries. Once the leaves get older, the nectaries don't secrete as much on the leaves. And so probably the sky is pretty much unburdened by being attacked by visiting ants. But the florivory and the seed predation are significant problems for this plant. And part of the problem of understanding it and investigating is that herbivory can be very variable between years. So in some years, and this is a year like that, I was just at my site last week and the plants look roughly like this. They're really nailed. In other years, there's not as much herbivory. I don't know whether there's a genetic component to it, but certainly there's a year-to-year variation. And that makes it hard to plan ahead for experiments because you're never quite sure whether the herbivores are going to be present or not. This is Galen, and he's the guy who took on the task of trying to document the ant interactions with extrafloral nectaries. And I'm going to just give you a fast look at the questions he asked and the data that he got. And this work was done in 3 sites at Horseshoe Canyon, also in Molino Basin, and in Mexico, in San Javier, Mexico, in Sonora. And part of that was because we were asking in part a question about whether or not the extrafloral nectaries were as effective here as they might be in the center of the range. As I told you, plants at the periphery of their ranges are experiencing different selective pressures. And so one possibility was that these plants really got nailed by herbivores in Mexico, but maybe at this end of their range, their herbivores were not still here. So we were wondering if there might be some sort of a latitudinal gradient in that process. So what are the ants present? How do the ants respond to herbivores? And is there any evidence that they actually protect the plant? These are Galen's data about the ant species present in the 3 different habitats. And you can see that there's a lot of similarity in the ant fauna between these 3 locations, probably because they're very similar environmentally and in vegetation as well. So there are a suite of ants, but none of these are obligate mutualists. None of these are require the nectar of erythrina to survive. They're simply facultative mutualists whatever ant happens to be nearby a plant and finds the nectaries will take advantage of it. So the possibility is that some of these guys are actually pretty good at defending the plant from herbivores, but some of them are not. We did 24-hour censuses in all three environments to try and track the ants on the plants. And so this is just one of the data sets from the Horseshoe Canyon site. One of the things that really surprised me, I just didn't even think about the fact, would be that the ant fauna changes over between the day and the night. So the ants that are present in the plant during the daytime are not the same ants that are there at night. And they're different species that appear, and the abundance of the different species changes over the day. So this is 7 in the morning. This is 7 at night. And clearly there's a lot more ant activity in the evenings when the temperatures are low. And that does seem to be when a lot of the nectar is being secreted as well. To test whether these ants would be potentially good against defending the plant against predators, we did some feeding experiments. So we bought these little waxworms at the pet store, and we put them on the plants and looked to see what the ants did. And it's kind of gruesome, but on the left, there's no caterpillar. And on the right, under that mass of ants, is a caterpillar to which the ants have recruited and are happily consuming. So this wasn't the case for all ant species, but at least one of the ant species that we were finding on these plants was in fact potentially an anti-herbivore actor in this scenario. And then finally, we did some exclusion experiments where we took branches on the same plant, and in some of them we excluded ants from the branches by coating the stems in a gooey substance called tanglefoot, so that on some branches the ants could not patrol the inflorescences, and on other branches the ants were free to move up and down the stems. And where there were ants, and the results are more subtle than this. I'm making it as simple as possible, but where there were ants, we saw lower events of herbivory on the inflorescences. It's not that there was none, but there were lower probabilities on those populations where on those stems where the ants were allowed to visit. So in general, there seems to be a facultative mutualism going on here between the plant and a variety of different ant species, in particular chromatogaster, which is the one that's the best at attacking the prey items. The different ant species vary in their ability to defend the plant, but in some cases they do seem to be making a difference. And part of the problem of really pinning this down is just that the ant abundances change between years, the level of herbivory changes between years, and all of those factors are going to affect the ant-plant interaction. But in addition to knowing that there are ants on these plants, the additional question is that there's a lot of nectar being secreted here. So we've got all these extra floral nectaries on this plant secreting nectar day in and day out up through the time when the flower is pollinated. And that's a lot of sugar and a lot of water to be producing for a potential visitor who may or may not be doing very much to help the plant along. So we really wanted to try and quantify the cost of this potential mutualism to the plant. And Nikki Gustafson, one of my students who graduated in 2014, took on this project, and it was quite a challenge, but we got some really interesting results. So we wanted to ask how variable is extrafloral nectar production? Is there variation between plants? Is there variation between inflorescences in a plant? Are there potential genetic bases to this such that there could be an effective natural selection on this? And what's the actual investment that a plant puts into producing extrafloral nectar? So the first question was how to measure it. In the desert, extrafloral nectar doesn't stick around very long. In the daytime, you cannot see extrafloral nectar on these nectaries. You can see the ants there who are apparently sucking up anything that gets secreted, but there's no standing nectar to measure. We devised a method by which we bagged the plants to allow the nectar to accumulate. So we wanted to ask, is there differential nectar secretion among very small stage 0 and stage 1 buds? And does the nectar increase as the flowers get older? Or what's the timing of nectar production by this inflorescence? So we scored the plants by different stages. Stage 4 is the first day flower, stage 5 is the second day flower. And then we bagged the plants in the evenings after the sun went down with plastic bags in such a way that they weren't going to be hit by direct sun, and they would simply be, we would be keeping the ants off the plant during the course of the night. And we let the nectar accumulate, and then the next morning we got up very early and went out and uncovered each of these plant bags and looked at the nectar that had accumulated. And we had to do it before the sun hit the plants, because if the sun hits a plant in a plastic bag, it immediately gets cooked. So we spent a lot of time doing this. We were originally going to use a micropipette to gather the nectar and measure its volume, but we couldn't do it because when we took the bag off the plant, the nectar started to evaporate instantly. So the way we quantified this was by taking photographs of each of these inflorescences as we took the plastic bag off, and we put a scale in each photograph, and then Nikki went in with a photo program and measured the diameters of each of the little accumulated droplets on each of these flowers. And then we calculated the volume using a conversion in order to quantify the amount of nectar being produced by flowers at each stage. The remarkable thing is that when you bag these flowers, you get visible nectar accumulation. These are extrafloral nectaries, but they're creating droplets of sugar solution whose concentration is roughly the same as the floral nectar. And even plants that have no open flowers are producing huge amounts, huge relatively speaking, amounts of nectar, even on the smallest flowers. So the result of our efforts was to try and identify where the sources of variation were in this process. We started by I took out the data slides to make it easier, but we started out by measuring the sizes of the nectaries, and we found that the sizes of the nectaries on the tiniest buds were almost full size. The nectaries themselves did not enlarge very much as the flowers developed. The flowers got bigger, but the nectaries were pretty much almost full size when they first emerged, suggesting that getting some nectar out there on your inflorescence early is really important. So the nectaries are virtually full-sized in the smallest buds. Plants differed among plants in the total amount of nectar that they produced, but the nectar secretion did not differ between the buds and the smallest buds and the buds that are beginning to produce flowers. So the actual volume of nectar was significant even on the tiniest buds. And when we looked at this is a graph, this is a sort of the heuristic take home. If you look at the average nectar production during the life of a flower, we combined the extrafloral nectar that was being produced. Remember, that flower was a bud for about 3 weeks before it became a flower. And for that entire 3 weeks, every day, it was producing extrafloral nectar. And then during the 2 days that it was a flower, it was producing floral nectar. So we wanted to know what's the comparison between the nectar production by the extrafloral nectaries and the floral nectaries. And what we found was that plants are investing very heavily in the extrafloral nectar as well as in the floral nectar. The floral nectars are shown in the sort of greenish bars. The extrafloral cumulative, that's cumulative nectar over the life of the bud, are shown in the dark bars. But when you add together those tiny little amounts of nectar for 3 weeks, you find that the energy that the plant's putting into defending itself as a bud is almost equivalent to the energy that it's using to try and recruit pollinators. To me, that's a really interesting conclusion, especially when we aren't even really sure what the herbivore situation is year to year. And there are many years in which we see very little damage to the flowers for which this nectar is presumably being produced. So this may be an example of a trait that has selective value in some environments, but it doesn't have such a high selective value here at the margin of its range. Nonetheless, it's pretty dramatic to see this kind of investment. Okay. And then just briefly, the sort of unfinished part of this work is to try and pin down the genetics. And I'd really like to know more about the pollen movement in the population, the paternity of seeds that are being produced, and the reproductive fitness and how it changes from year to year. And we've made some initial starts. We've genotyped the adults in our population at the Amaranth, but we still have a lot of individual seeds to do the genetics on. But the questions involve things like, how far do birds fly between plants? Do they move between populations typically, or do they tend to fly within a single population? Are there some plants that are super moms or super dads and that are contributing either lots of pollen to the offspring or lots of seeds to the offspring? And how much does this change from year to year? The plants themselves can actually change size from year to year, given the fact that they can die back from frosts, they can be burned. So a plant that's gigantic in 1 year may in fact be reduced to almost nothing in the year next year. And so tracking the genetic components of this is incredibly interesting to me. It's just something that we haven't yet quite realized. What we do know about the genetics is this, that in testing 44 seeds genetically, 3 of those 44 seeds were definitely pollinated by fathers from outside the population. Within a single fruit, we found that there was more than one father that contributed to the genetics of the seeds in a single fruit. For 17 of the seeds of these 44, where we had a pretty small estimate of potential fathers, the mean pollen movement in the population was between 36 and 53 meters. And as I mentioned earlier, only one of those 47 seeds could have been self-pollinated. So what that tells us is that selfing is not happening regularly in this population, or at least the self seeds are not being matured. So outcrossing is definitely the rule in coral bean, despite the opportunities for selfing that are presented by its biology. So the take home is just that if you're a nerd, and I obviously am, as you've heard for the last hour, if you're a nerd about this kind of stuff, these plants live pretty interesting lives. This is plant 1342 with my student Mia in front of it in 2019. And the plant was doing relatively well. It had quite a few flowers on it. In 2020, it was doing exceedingly well. It had lots of great fruits on it. But in 2021, there was a fire. The plant was burned back and a chunk of an oak tree fell on one end of it. And this year it had very, very few flowers on it. So the vagaries of the population change from year to year. But through it all, Erythrina manages to still hold its place as one of the loveliest plants in the Arizona flora. And I have lots of people to thank. The people in blue are my students who assisted me in various summers of research. People in red, the organizations in red, are people who have supported my work in very substantial ways. And the green is my funding, and I appreciate your attention. I'd be glad to take any questions.
Questions. So if folks would like to type in questions on the Zoom, feel free to pull up the chat and do so. I'll be reading the questions for Lyn to answer. People in the room, feel free to raise your hand. I'm going to try and facilitate Zoom questions first. This one's from me. Do you have a theory as to why there are so few herbarium records from the Chiricahuas, given the numbers of plants you have seen there? And how did you find out about the Chiricahua populations without there being herbarium records?
Well, I think there are probably records that haven't been geolocated yet, and so they don't show up on the map. And since I manage the herbarium at the Southwest Research Station, I know we have sheets that are there. We've got them in SciNet, but they're not geolocated, so they just don't show up. I heard about the population from a longtime local resident who told me there were plants in Horseshoe Canyon, and he was right. So.
Did you observe that most burnt plants recovered, or has there been significant mortality from the last year's fire?
That's a good question. And I would have to say that there's definitely more mortality this year after the fire than in previous years. The fire was fast and it was damaging to last year's stems. But by the end of the summer, most of the plants in my population had sprouted from the base again. So even though their fruits and their leaves and their upper branches were gone, they all sprouted out in lots of new sprouts. But there were some plants that got cornered or were growing under a dead oak tree that went up in a conflagration. And so there clearly is a signal of mortality from the fire, but most of the plants came back.
We have a question. What does fully pollinated mean?
Fully pollinated would be that enough pollen is delivered to the stigma that all the ovules are actually fertilized and develop into fruits. And so a plant has 10 to 14 ovules that are potentially seeds, but they don't all get pollinated if there's a small pollen load that's delivered, if the bird doesn't manipulate the flower in the right kind of way, if there are genetic compatibilities between the pollen and the flower. Fully pollinated would mean 10 to 12 nice seeds that would be great for my genetic work, and half pollinated would be a couple of seeds.
Is the distribution of coral bean reflected in the distribution of hummers with 28 millimeter bills?
No, clearly not in Arizona, because we don't have, I don't think even Rivley's has 28 millimeter bill, but there are certainly larger bills in Central America and in Mexico. And so that may be one of the reasons why you get better fruit set for the flowers you have down there. I don't know for sure, because you can go out there and watch for pollinators, but pollination is not very common. On average at Amarant, if you watch a set of plants for an hour, you'll probably see one bird. So it's not like these plants are living in a hummingbird bonanza. In fact, when we first started this work, the first year I started working in Horseshoe, I had my whole summer agenda planned. I had a hillside covered with plants. We're going to put numbered tags in front of each one. We're going to sit under the mesquite tree with beverages and watch with our binoculars as the pollinators moved from plant to plant. And the first day we went out there, I'd sold this to 2 students who came all the way out here to do this for the summer. First day we went out there, we didn't see a single bird. The second day, we didn't see a single bird. And that whole summer, we saw about 3 birds. And we were out there every day for 5 to 6 hours. Now, we were in one place and there were plants in lots of other places. The birds could have been elsewhere. The birds don't visit very often, except in some years, there are more. And Horseshoe is a habitat where there's no free water in the summer. The only water is the cattle tank at the base of the valley. And so it may just be a situation where there aren't many birds because the habitat, for other reasons than food resources, doesn't support the birds. So.
Have you tried doing any self-pollination and then begging the flowers to test selfing?
Yes, and that the data that we have about whether or not seeds can be set on first day or second day flowers all comes from self-pollination. So we've done a bunch of self-pollinations early in the season, and we found that plants could be pollen donors or egg parents in both first day and the second day, selfing and outcrossing. And it was approximately under controlled seed sets, the seed set was about the same with selfing and outcrossing for the fruit set. Now, whether or not those seeds would have had genetic problems or not, I don't know. But when we selfed, we did get seed set.
I'm not entirely sure how to interpret this question, but is any similar information known for plants from the center of their distribution? I'm wondering if that means if has similar research to what you're doing been done in the center of the population of Erythrina flabelliformis?
Not that I'm aware of. We've worked as far south as Alamos, but we've only been able to stay for short periods of time, so we haven't been able to do the same kind of manipulations. And part of the problem down there is that the reason it's so nice to work on Erythrina in Arizona is because it's short. I can reach the flowers, I can do crosses, I can bag plants, and it's not a problem. I worked on an erythrina in Panama, and I had to climb a 10-foot ladder to get to the flowers. And I fell off one time, and I decided this was not my future research project. So I was delighted when I saw that Arizona had short erythrinas, but the ones in Mexico will be much less easy to work on.
And a second question from the same person: how do big red seeds move away from the parent?
That is probably the single biggest unanswered question. I do not know what disperses these seeds except gravity. And you can go to Horseshoe and you can look up those cliffs, and there are erythrina plants flowering at the top of those cliffs. And I don't know how the seeds got there. I have found seeds and pods in pack rat middens. I don't know how frequently that happens, but I suppose it's possible that an industrious pack rat could truck them up the hill. For a while, actually, my hypothesis was that erythrina was brought to Earth by aliens, and it was immortal, and all the plants that were here were going to be here forever. But I haven't been able to get very much evidence to support that hypothesis. And I have subsequently seen seedlings, but how they move uphill, I do not know. Everyone says, I mean, they're red and they dangle out of the pod when the pod is mature. So that's a classic tropical dispersal by birds. It's a classic mechanism for birds to go in and pluck seeds. There's no reward, there's no aril or fleshy fruit around the seeds. So it would have to be a kind of a, sort of trick dispersal. You'd have to have a bird that was expecting a reward, but then was being tricked into taking seeds that weren't rewarding. For a while, I harbored the hopeful hypothesis that maybe they were dispersed by the extinct parrot that used to live in Arizona. But in fact, parrots in the tropics mostly are destructive on the seeds. They don't really eat the seeds and pass them. They chew them up and spit them out. So I do not know how they're dispersed, except if a pod is dragged by a rodent of some sort. The seeds are toxic. But if you had a fast passage time through your gut, of course, then you wouldn't get any of the nutrients out the inside if it just passed through. So it's the central question of my existence, and it's what I lie awake dreaming about at night.
And does anybody in the room here have a question? I can hand you the microphone.
Yeah.
The seeds are toxic, right?
They are reputed to be toxic. I have no personal evidence on that.
Oh, so you haven't investigated what
I
what the
haven't.
mechanism is?
Although Rex just showed me a comment that somebody made on the chat about some ranger at Saguaro National Park who was told, but he was naive and he was told that they were edible, and he ate one and got sick, quite sick. But I know I'm only reading what somebody put in the chat. They're supposedly poisonous. Ethnobotanically, the indigenous people did use them in small doses for medicinal purposes as emetics and so forth. But they were also reputed to be used as ways of getting rid of people that you didn't really want to have around. You'd mash them up and put them in their food. This was reported by some of the informants to Austin, or to one of his sources. There's a Tarahumara legend that the two giants that took over their land and pushed them out early in their history were eventually dispatched by being poisoned by erythrina seeds so that the Tarahumara could reoccupy their land, if I understand the story correctly. But the seeds are presumably poisonous. Yes, but you had another part of that question. It's a neurotoxin, apparently. It has curare-like effects. There are there are allusions in the literature to erythrina being potentially hallucinogenic, but although I've seen that cited, I haven't seen any demonstrated evidence. And there's always a problem because the coral bean seeds are very much like sophora, the Texas mountain laurel, which are hallucinogenic, but there are sometimes people who would confuse them. And in fact, the Apache and others who use seeds in this area often used both sophora and erythrina in the same decorative fashion, tying them onto fringes and so forth. Yeah, they're supposedly poisonous. Apparently the leaf material is not, and the flowers are eaten in Mexico. They're apparently stir fried with eggs. It's supposed to be very good, but I haven't tried it. But the seeds are toxic. And if you go into Google Scholar and Google erythrina, most of what you get is biochemical citations of all the alkaloids that have been extracted from their seeds. Okay.
Oh, wait. But I don't have a question. Lynn just looked at me and but I can imagine a question. Looking at that bloom there, I was wondering would a hummingbird visiting such an inflorescence, how many flowers would it typically visit there? Does it,
Well-
you mean that also temporally, I mean, flowers from all different times?
Really good question. And it's part of the whole mix. The hummingbirds do not stay very long at any one flower. They come in, they visit maybe 3 flowers on an inflorescence. They might go to another inflorescence and visit 2 flowers and they're gone. So it's not like bees where you have a spike-like flower and the bees land on the bottom and meticulously climb all the way up to the top, visiting every flower on the way. The hummingbirds are very erratic in their visits. And they move around a lot. And you wait an hour to an hour and a half to see that.
That might have something to do with the outcrossing.
It could certainly have something, it would promote outcrossing for sure. It would also potentially mean that you wouldn't have much seed set. You'd have, on the other hand, one good visit can set fruits. And one of the things I didn't really talk about because it's hard to explain, but if you look at an inflorescence with maturing seeds, the seeds tend to be clumped. So that it's like the flowers in 1 or 2 fascicles that were all flowering at once have produced fruits. And then there's a gap. And then the next fruit is a couple of days later. So I'm interpreting that likely to be the result of a hummingbird visit where he or she visited 3 flowers in a row and then took off. But if you do a Poisson test on them, you find that there's clumping in the arrangement of seed pods on a stem.
All right, cool. Well, thank you everyone so much for being here on Zoom and in person. Thank you so much, Lynn, for your excellent presentation. That was really entertaining. Thank you so much. Everyone have a fantastic evening, and don't forget the field trips I mentioned at the beginning of this meeting, as well as our Waterman excursion on October 22nd. That's going to be a big hoopla, and everyone should come out to see the incredible work that John Schuring and our conservation folks and all of the contributors have done there. So thank you, and hopefully we'll see you next time. Have a good night. Running around like crazy.